Transporters. Proteins in disguise.
Wait. Hold up. These things aren't even disguised. There's a picture of them right there. Right there. Seriously, who writes this stupid script? Hello everybody, and welcome to another BMSC 200 video. Today, we are going to be talking about solute transport across membranes. So here's a reminder of what our membrane is. It's made up of phospholipids. We have a hydrophilic head group, and we have a hydrophobic tail. These lipids have that business up front, party in the back look. Anyways, we have three main ways of transporting solutes across membranes. We have simple diffusion, facilitated diffusion, which uses channels and carriers, and active transport. And there are two forms of active transport. There's primary and secondary. So we're going to focus on simple diffusion first. This is the basic pumpkin spice latte loving UGG wearing transport system. Something starts out, it moves in. It goes out and in. And out and in. This doesn't require a carrier. It's non-saturable, meaning it's unlimited. We go from an area of high concentration to low concentration, and it requires no energy. Perfect for those lazy university students just like you. Seriously, go outside for once in your life. Typically, nonpolar gases, so gases like oxygen and carbon dioxide and other hydrophobic molecules, can directly cross the membrane down a concentration gradient. Think of yourself breathing. You breathe in oxygen, you breathe out that beautiful carbon dioxide. They have to pass through a membrane on your lungs. Breathe in, oxygen passes into your bloodstream. Breathe out, see you later, CO2. If you are unable to do this breathing exercise, consult a doctor immediately. You are a robot, or better known as a
Transporters. Proteins in disguise.
Now let's talk about facilitated diffusion. It's important to note that the transporters help lower the activation energy barrier of molecules moving across the membrane. We are transporting a polar molecule across a non-polar part of the membrane in this case. The hydrophobic tails of phospholipids hate polar molecules, which is why we have the transporter, to prevent polar molecules from interacting with the non-polar tails. Wait a minute, is that a
Transporters. Proteins in disguise.
I'm just gonna keep putting this meme in this video. You guys tell me when it stops being funny. Now, we have two main types of
Transporters.
No. That's enough. We have two main types of transporters in our body. Channels and carry. I give up. Carriers, thank you, in your body. Channels are membrane pores that can only transport molecules and ions down an electrochemical gradient. They're super fast because they bind their substrate very weakly. Their speed is proportional to substrate concentration. So basically, more substrate equals more speed. And here's a quick demonstration of what happens with facilitated diffusion using channels. I was gonna make another joke here, but I can't think of anything funny, so...
Transporters.
Now, carriers have a rate-limiting step where the substrate binds to the protein. This is identical to the enzyme-substrate complex we saw earlier in class. Because of this fact, carriers are typically a lot slower than channels. Carriers can work both with the electrochemical gradient and against it. Here's a quick demonstration of what happens with facilitated diffusion when using carriers. On top of channels and carriers, we also perform facilitated diffusion with coupled transport. Now, for some of you, you're thinking of your significant other. You're thinking of your couple relationship and how much you love them and how beautiful their eyes look. To those people, I say, bah humbug. To those of us perpetually fearing our loneliness, congratulations! You can finally be part of a couple for once in your life. With yourself, please don't make that comment dirty. We have three types of transport. Uniport, where one molecule goes by itself alone. Symport, where two people walk through together. and antiport, where two molecules come and go from opposite directions and look back at each other like, dang. These are done to prevent changing the electric potential across the membrane. Finally, we have active transport. These are the jocks of transport, the guys that only talk about how swole they are or how much they can bench press. Primary transport is driven by energy, which is ATP, of course. Secondary transport is driven by an ion gradient. The example in your notes is the sodium-potassium ATPase transporter for primary transport. Now, most people forget which goes where. I'm about to give you some of that sick knowledge. Knowledge. K goes in your DMs, NA is like, I'm out of here. That's the thing you remember. Nobody wants to hold a conversation with you, and after you text them, they're like, nah, I'm out of here. It's kind of like my love life. Now, how many K? You got 2K, and you got 3 NA. Here's a fact that many of you might not know. I'll speak some biochemical truth in your life, yo, faux show. Now on your way in, you got that sweet 2K. And on its way out, you got the tray NA. Now that's a fact you'll never forget. How true is that? That's enough knowledge for today. Let's finish this rap. I shouldn't ever rap. Here's how the mechanism works. 3 sodium binds. ATP phosphorylates the protein, which causes a conformation shift and it flips to the other side. We then release our 3 sodium and the 2 potassium slides in. The protein releases the phosphorus, again causing that conformation shift. This time it's opening to the inside of the cell, and shabam, we have 2K on the inside of our cell. Now in secondary transport, remember that we had said this is driven by an ion gradient. Ions go down their concentration gradients, molecules go up their concentration gradients. Now, primary active transport has to happen first before we can have any secondary active transport. This is because we need an ion gradient, which primary transport does with the sodium-potassium transporter. The example in your notes is the lactose transporter, where lactose goes inside with hydrogen. Hey, why do cows have hooves instead of feet? Because they lactose. I hate myself. The other example given is with glucose uptake. Glucose plus 2 sodium go into the epithelial cell. They are then joined by a rogue sodium, making 3 sodium. They swap with 2 potassium. We know the rap, I'm not playing the audio file. Glucose is then taken up by a uniporter. And this is how glucose enters your cell. Now that you've learned all about carriers and channels, go ahead and subscribe to this channel. Or don't, whatever, it doesn't matter to me. Anyways, thanks everyone for watching. Channel your sodium and potassium to crush this final. Every day is better with biochemistry. Have fun, love life, and I'll talk to you all in the next one.